Brown fat, a special type of fat cell, plays a crucial role in maintaining body temperature by converting calorie-rich nutrients into heat. This process, known as thermogenesis, helps protect against excess weight gain and metabolic disorders. Unlike white fat, which stores energy, brown fat actively burns calories to generate heat, making it a key player in energy expenditure. Understanding how brown fat functions can provide insights into combating obesity and related health issues, as it offers a natural mechanism for burning calories and maintaining metabolic health. Brown adipocytes, the cells that make up brown fat, are particularly adept at attacking fat stores. When the body is exposed to cold, these cells extract fuel from stored fat to produce the necessary heat, a process that requires a significant amount of calories. This activation of fat-burning cells can lead to weight loss, as they consume stored energy to maintain body temperature. Professor Alexander Bartelt from the Institute for Cardiovascular Prevention highlights that individuals who regularly expose themselves to cold conditions tend to be thinner and less susceptible to diabetes and cardiovascular diseases. An international team of researchers, led by Professor Bartelt, has uncovered a new mechanism that enhances the respiration and metabolic activity of brown fat cells. This discovery, published in the MBO journal, reveals how brown fat cells can be more effectively utilized against metabolic diseases. The researchers identified a molecular switch that increases the efficiency of brown fat cells, potentially paving the way for novel therapeutic approaches. By understanding these mechanisms, scientists hope to develop strategies that harness the power of brown fat to combat obesity and improve metabolic health. Brown fat cells are rich in mitochondria, the cellular powerhouses where respiration occurs. A key component in this process is uncoupling protein 1, which allows heat generation instead of producing adenosine triphosphate, or ATP, the usual energy currency of cells. Professor Bartelt explains that the high metabolic activity of brown fat cells influences ATP production, and this process is regulated by cold exposure. The researchers, in collaboration with Brazilian colleagues, identified inhibitory factor 1, which maintains ATP production over thermogenesis. When temperatures drop, levels of this factor decrease, allowing thermogenesis to proceed. Cold exposure plays a significant role in stimulating thermogenesis in brown fat cells. The study found that when inhibitory factor 1 is artificially increased, it disrupts the activation of brown fat in cold conditions. This finding was confirmed through experiments with isolated mitochondria, cultivated cells, and animal models. Dr. Henver Brunetta, who conducted the study, notes that while this discovery is a crucial piece of the puzzle in understanding thermogenesis, practical therapeutic applications are still distant. Most people underutilize their brown fat, leading it to become dormant, highlighting the potential for targeted activation. The study's findings suggest that there are molecular switches that can enhance the function of brown fat cells' mitochondria. Professor Bartelt and his team aim to build on this discovery to find ways to restore mitochondrial fitness in white fat cells, which are abundant in most individuals. By leveraging the data from their research, they hope to develop new methods to activate brown fat and improve metabolic health. This could lead to innovative treatments for obesity and related metabolic disorders, offering a promising avenue for future research and therapeutic development.